Thank you. All right, I want to welcome everyone to the this journal club hosted by the Center for Translational Social Neuroscience at Emory. And we're delighted to have Karl Deisseroth here to talk about his new book, which we'll get to in just a minute. First, I'd like to introduce Karl. So Karl is a Howard Hughes investigator and also professor of bioengineering and psychiatry at Stanford University. And he's probably best known for pioneering one of the most influential and powerful techniques in neuroscience in the last couple of decades, and that's optogenetics. Optogenetics has allowed him to gain incredible insights into the working of the brain and the mind, and not only him, but it's one of the most prevalent techniques in neuroscience today, in all aspects of neuroscience, to understand how the brain works. And so he's really had a really important contribution. He's one of the most well-known neuroscientists in the world today. And so I think we can go ahead and get started. Thank you so much, Karl, for coming and speaking to us today.
Oh, thank you for the invitation. This is great. It's great to see everybody. Wish I could be there in person, but hopefully we'll get that done at some time in the not too distant future.
Yeah, I hope so. But at least this gives us the opportunity to do things like this, which I think is great. So, Karl, we have a diverse audience here. So some people are neuroscientists, bioengineers, but also undergraduates and members of the public. And I wonder if you could just first just give us a brief overview of what is optogenetics.
Sure. Yeah, optogenetics. This is for the. I'm glad there's a broad audience here. That's really what is so exciting, to reach out and connect with people from all backgrounds. And so optogenetics, just from the perspective of how you use light as a scientist and as an organism, we usually think of light as a way of collecting information. Optogenetics flips that around. We use light to make things happen. We use light to play in information and cause particular groups of cells or even individual cells to turn them on or off. The electrical activity in those cells, turn them on or off in real time as a complex animal carries out complex actions. And this is great because then you can decide what actually matters, what's causal, what can and does matter for sensation, cognition, action, if you're a neuroscientist, but you can apply this method to any kind of cell. And the way we do it, extremely briefly, is most cells in the brain don't respond to light. It's very dark in there. There's no reason for them to respond to light, which is great because any little bit of photosensitivity, light responsiveness you could add is on top of a zero background, and it gives you a very high signal to noise. And so what we do is we take genes from microbes that are light activated ion channels that move charged particles, ions, across the membrane of cells in response to light. Just little molecular machines that receive a photon, open a little pore, charged particles rush across the membrane. These come from microbes that do that for their own reasons, but if you take that gene and you put it into neurons, you can make that neuron electrically responsive to light. So that's the whole concept, and it's very useful. Took about 5 years to develop all the parts between about 2004 and 2009 or so for the bread and butter optogenetics that's widely used today. Mm-hmm. We use specially designed viruses that help us get these microbial genes into the right cells. We use special light guidance tricks like fiber optics to get light into the brain. And these days we're even using holograms, playing in 3D patterns of spots of light so we can target individual cells even, and turn on dozens or hundreds of individually specified cells in or out of synchrony in any pattern we like during behavior. So that's the essence of it. It's a lot of fun to do, and as you say, it's a lot of interesting insights have come from around the world.
Yeah, you were awarded the Lasker Prize, 2021 Lasker Prize, along with 2 other colleagues for doing just what you just talked about, right? Developing this light-sensitive channel.
Yeah, and I'll mention their names too, Peter Hagemann and Dieter Osterheld. These are amazing scientists, friends, and colleagues and collaborators. They're both in Germany, but they were they worked on these basic microbial proteins themselves. And together with Peter, we also discovered a number of these new ones, and then we worked on the crystal structures of these proteins to understand how they work and re-engineer them for new kinds of functions. So it was a really nice way to recognize the sweep from the pure basic science to the structure, to the discovery, and to the application.
Now, for the aficionados of optogenetics, can you tell us what are you most excited about coming down the pike? What's the future?
Well, you know, there's been a progression over the last 10 years or so. Starting in about 2012, that's when we first were able to control single individually specified cells. And that was a big leap forward, but it took about almost 10 years for us to get really facile with that, to be able to control during, let's say, the behavior of a mouse, multiple individual cells all at the same time or with different timing. And now we can do that very in a very straightforward way. So for example, we can take a mouse and we can listen in or watch in, we can see the natural pattern of activity using activity reporters within, say, visual cortex of a mouse. We can find all the cells that respond to, let's say, vertical bars instead of horizontal bars. And we can find all those cells that are naturally involved in reporting vertical and not horizontal bars. And then we can play in a pattern of activity. We can pick out those cells for optogenetic stimulation. We can stimulate just those cells, the vertical cells, but not the horizontal cells. And we can make a mouse act in every way as if it's truly seeing something, truly seeing vertical bars in terms of behavior, both what it does, the variance with which it does that, and the internal representation is also naturalistic too. If we look at all the cells that are recruited, it's naturalistic as well. So this is kind of interesting. We now have the ability to effectively provide a naturalistic single cell resolution percepts, if you will, in an animal, and keeping the brain intact while we do this and do increasingly complex versions of this is a really exciting prospect. And so that's where things are going, getting to sort of the completeness of individually specified cells, but across a brain. And we're doing this in mouse, we're doing this in zebrafish, where it's easier to get to completeness, the much smaller brain, many individual cells still accessible. And so we're headed down an interesting path where the ability to specify what happens is really reaching the potential for completeness. It raises all kinds of interesting computational problems, analytical problems, even philosophical questions. But it's an exciting progression.
I mean, could you conjure up a memory of your grandmother or something like that?
Well, as we look at we stimulate some cells and we look at the responding cells, and indeed they do resemble the patterns and the dynamics of what's naturally recruited. And so as far as we can tell, that sort of thing is possible.
That's very cool. So we're here today to talk about your book, and congratulations on this book. I just finished it, and I highly recommend this to anyone who, whether you're a scientist or a scientist, a psychiatry resident or a psychiatrist, it has something for everyone if you're interested in evolutionary biology. So first, I guess, why don't, Karl, you let us know a little bit about how you got to this book. How did you go from being working in the lab and being a psychiatry resident to this book?
Yeah. It's like many things in life, a lot of twists and turns, but deeply rooted, actually, even in childhood. As a psychiatrist, I often can look for those deep roots in childhood, but I always loved words. I loved writing. I loved literature. That was my first passion, actually. I wanted to be a writer. When I went to college, I signed up for a creative writing course first thing, and I loved it, but I got more interested in science later. But I always had this love of words and how they stir feeling, and was curious about that. And actually that led me to neuroscience in many ways, and thinking about the cellular basis of feeling and emotion, which many of us think about and care about a lot. And so I did an MD PhD program, and I was thinking I had to go to neurosurgery, and that way I could talk to people while building some kind of new way of studying the brain, and ask people about feelings while studying how the brain was manifesting those sensations. But then, I did in medical school, you do these clerkships or rotations, and I did my psychiatry rotation, which was required, and I was just blown away by it, how much need there was, how much suffering there was, but how interesting it was also. And so I switched paths completely, did a psychiatry residency, also at Stanford. And I had a transformative experience just about 20 years ago that made me do that switch. It was seeing a patient on the psychiatry ward, and really the stories in projections in the book are, each of them is from a really a transformative personal interaction with a patient. There are human stories of people who are coming to my care in these very altered states. But over the years, over the 25 years or so that I've been doing this kind of thing, there are a lot of individual patients. Which are the relevant stories? How do you distill down? What's the way to really make it happen? And the first one was just about 20 years ago. It was right after 9/11. This really got me down the path of writing the book. I had a patient who was retirement age, had never had any psychiatric issue in his life, nor in his family at all. And he was flipped into mania. This is the elevated pole of bipolar disorder by 9/11. He was actually vacationing with his wife, and about 2 weeks after 9/11, he had no greater connection than anybody else in the country to the events of that day. But he developed a classic mania, stopped sleeping, incredibly elevated energy, mood, goal-directed activity, words coming in an unstoppable, rush, and it was all very aligned with the nature of the global situation. And it really made me think quite a bit about, why we as humanity, as the human family, we have these susceptibilities to psychiatric illnesses, what they really mean, what they reflect about our genetics, our evolution, who we are as people. And I wrote a story, I wrote a book about it. I wrote a chapter, what became a chapter at the time, but I just sort of kept it with me. And I over the ensuing years, I had some other thoughts, and I wrote them down, and sort of reactivating the love of words from my childhood. And then in 2013, the New Yorker did a piece on me, and I happened to mention that I had done this to the writer, and the writer made that part of the piece. And then I got contacted by agents. This is still almost 10 years ago. So I'm a slowly developing writer for sure. But they contacted me and said, hey, can you write something? And I shared the chapter, and they were interested, but it took me a number of years, busy with other things. But then it really picked up steam in 2017, 2018, and I got it done. And the goal really is to bring these thoughts and ideas to everybody, to the public. What do these altered states mean? What are they like? I wanted to share them, reflect them in the real words, the real feelings, and the inner sensations of these patients. And so in the different chapters, different stories in the book, I use very altered language. Very elevated language, very elaborate for the mania story, but also in case of schizophrenia or psychosis, very fragmented language, and in depression or dementia, very constricted language. And so it was the goal to try to reflect the feeling, the inner feeling of these states for people. And so that's how it came together. And it's to be able to circle back that far in time and connect with your original passions in life is a pretty interesting process.
Yeah, what I love about the book is, each chapter, it starts with a disorder in a patient's and your experience with that, and then goes on to the neuroscience and optogenetics. And so it really allows you to see, both aspects of your work, but also, as you said, you have a love of words. And so this is not just a science book. It's literary, right? It uses very interesting language. In fact, let's look at that first, that chapter that you were just talking about with the person with mania. I believe the person's name was you used was Alexander in chapter 2.
Yep.
Can you read a little description about from your time as the psychiatrist there?
Yeah, I will do that. This is, and yes, I do. The patient's name here in the book is Alexander. Of course, I changed all the details for privacy, but the symptom descriptions are real. And so I think it can capture, for people who haven't been in the presence of someone in mania or had friend or family or themselves experienced it, it's quite a very remarkable discrete state and really a stunning thing to see. The way these things present, though, it's when, this is not to romanticize it. This is a really serious situation these people get into, and they really, the process of mania can be very, not just disruptive, but life-threatening. But it's at the same time, it's this elevated state of charisma and optimism and exuberance, and so there's this tension there that is very interesting. Okay, so I'll read 3 paragraphs from early on how Alexander presented to me in the clinic. The first manifestations were physical. He nearly stopped sleeping, becoming fully alert and charged with life for 22 hours a day. Never the chattiest before, Alexander now could not hold back a vast volume of words that came out in a pressured torrent, turbulent and interjaculating, yet still coherent at first. The content of his speech changed too. He was saltier, charismatic, uplifting, and illuminating. Beyond language, his whole body was affected. Now ablaze in new youth, he was suddenly voracious and hypersexual. No old bull out to pasture, he was an organic being newly ready to react, to interface. His skin surfaces were functionalized and available. Life was lacier, compelling, alluring. Projects and goals came next. They were valorous and numerous, with a tinge of excitement, a sub-thrill of risk. He bought a new Dodge Ram pickup with a heavy-duty trailer hitch and extended cab. He ran all night, read books all day, and studied theory of war, writing pages on the movements of forces and reserves. A theme of self-sacrifice appeared and grew stronger. He wrote letters volunteering to join the Navy and was found one evening rappelling down a redwood trunk in the fog, training for war. He was breaking from his lifelong chrysalis, transforming into a newly emerged monarch. There had been a certain charm to the transition up to a point, but then he veered into thoughts of good, evil, death, and redemption. He had dwelt until this event in a sort of unperforated Lutheranism, stormless and modestly nourishing, with minimal connectivity to any other part of this life. Now he began to speak with God, first calmly, then frantically, then in a scream. Between these prayers, there were sermons for others in which he became irritable, swinging between euphoria and crying. Nearing midnight before admission, he ran from the house with his quail gun, throwing branches and bark at his sons as they tried to stop him in the yard. Police found him 2 hours later, at bay, in a thicket near a dry stream bed, ready to strafe the stinkweed. They took him and subdued him with the mundanities of medical legal incantations, all the energy still brimming up behind his eyes like tears. So that's how the patient presented, and you can see the nature of the state is really a remarkable thing, and the discreteness of it, how this appears. This is something that we're built for. Mm-hmm. And so then the questions become why, what does it mean?
Right. Yeah, so I was really interested when you're writing about this in the book. Then you go on and talk about, for this disorder, but also other psychiatric disorders, that maybe the genetics, the biological constructs of this are there is some evolutionary adaptive nature to them, and the genes that cause these disorders have not been lost, that maybe they have some function, and that only in certain cases and in certain contexts do they lead to these disorders. And you actually mentioned Joan of Arc in the context of this patient. Can you say what you were thinking there?
Yeah, so I, with a big caveat that, and I say this in the book, it's usually not wise for psychiatrists to try to diagnose historical figures. It's also very tempting and illuminating to consider people who were in these altered states in history, what might have been going on. And the reason is to understand why we have these states, why they've persisted. And there are many things that in our construction that we know are a little bit harmful, but also a little bit helpful. The trade-offs happen and all through genetics and evolution. And could psychiatric illness be one of those cases? So, Joan of Arc was really interesting because, very often you'll see people talk about her altered state, and they'll use framings of psychosis or schizophrenia because she was, which I don't agree with, and I'll tell you why. She had this, she did have this sort of altered state that started when she was in her teen years. She was came from a rural background, had no particular training in politics or war, and her altered state, the way it was framed was the mystery began to speak to her, was how it was described, and there was communication with non corporeal entities, and so people often try to frame this as psychosis, but she was extremely organized and goal directed. That's what sets I think her state apart. People with schizophrenia are very disorganized. It's hard for them to form coherent long-term multi-step plans that make sense in context. That's, in some ways, it's the heart of the disorders, the disorganization. But she was extremely goal-directed, had elevated energy, very elaborate planning, and it was aligned with a crisis at the time for France, which was an invasion from England. And the analogies with, the perception from Alexander of what was going on 9/11 really could not, be clearer. Here was also somebody who had no military training. There was a perceived threat to the country. And then flipping into a state of elevated energy, elevated planning, goal-directed activity, a prolonged elevated state that had some alignment with the moment. And this can look, mania can look like, have some superficial similarities to psychosis, because there is the elevated state is so elevated that it doesn't quite make sense. There can be a religiosity, as with Alexander, and probably with Joan of Arc too, that comes with mania. But that's just part of it. It's part of the possibility. What really is happening in mania is that it's a prolonged state of elevated energy and elevated estimation of the value of one's own action. And that can be very valuable, particularly.
That's a useful thing. We need that.
Yes. We're all familiar with, the beautifully orchestrated, fight or flight response that lasts for minutes. But we need prolonged versions of that too sometimes, and as beings, as social beings who build and construct and use immense energy over time to help each other and put together societies. A few minutes of elevated energy is not well aligned to the sorts of things that we do as social beings. And
we needed to fight and conquer others too, which is our ancestors did, and they were the most successful, right?
Yeah, well, yeah. So exactly. So you look at the trade-offs over time, and it's easy from that perspective to see how the potential to have an elevated state over a prolonged period of time could have been of adaptive value culturally and genetically. And so that's an example of the sort of thing that is explored starting from the human story and looking at genetics and optogenetics. We go into to the extent to which these elevated states can be modeled and created by turning up or down specific cells and specific circuits in the brain. And,
Yeah, can you say a little bit about that? Because in each chapter you do go into optogenetic experiments. Many of them were yours, but sometimes there are others, where it shows you how you get insights from studying these mice in the laboratory about the human brain.
Yeah, and so this bipolar or mania is a good example of this because, so first of all, it's incredibly genetically, heavily determined. It's bipolar type 1 disorder is one of the most genetically determined psychiatric disorders. And there are a few penetrant genes, but in general, despite it being so genetically determined, it's not like, sickle cell anemia where it's, you've got the hemoglobin and it's a very clear, single gene, single protein pathophysiology. With bipolar, it doesn't fall down that way. It's more of a patchwork of susceptibility genes that work together. And so the genetics hasn't illuminated what these elevated states are, but optogenetics has, because it turns out that's the more relevant level of inspection. There are cells and circuits in the brain that, when activated or inhibited, can create these states that look like mania, and links can be drawn from these cells and circuits to the susceptibility genes for bipolar. And a good example of this was work that we did in collaboration with Colleen McClung's lab. There was a exploration that she was doing. She was interested in the link between circadian rhythm alterations and mania. And that's a classic symptom. This decreased need for sleep is really remarkable in mania. It's not, they really need less sleep. Their circadian biology is fundamentally altered. They can maintain this almost not sleeping for substantial periods of time. Eventually things break down, but they really don't need sleep for a long period of time. And so Colleen was looking at the clock genes, which are involved in setting circadian rhythms, and she had found an interesting association with the dopamine neurons in the ventral tegmental area. And so using optogenetics and a great collaboration between our labs, a couple interesting things were found. First, that in these mice, that there was elevated dopamine neuron activity in these clock mutant mice. And these were mice that had very manic-like properties. They had elevated activity patterns and reduced need for sleep, and there was elevated dopamine neuron activity. And then we went in with her and the amazing students and postdocs in the lab, and we used optogenetic methods to turn up or down these cells, and found that we could add or remove these manic-like properties. So I think it's a valuable example to, all life we got to find, and all biology and all medicine, we have to find the right level of inspection. We can't understand disorders by thinking about individual atoms sometimes. We can't understand them by thinking about whole lobes of the brain sometimes. You got to find the right level of inspection. And for much of psychiatric illness, even though psychiatric genetics is very important and valuable, that doesn't always bring insight, but at the level of circuits, it does happen.
Right. So that's just one example of how you work in the neuroscience to talk about how you gained insights into the biology. And so here you were talking about dopamine neurons. How did you treat that person, Alexander, and were they appreciative of that?
Yeah. This is the interesting thing about clinical medicine is you can admire the problem, but then you also have to do something. And there's a human being in front of you who is, it's not working. And, with Alexander, it was, he had the gun, he was doing things that weren't safe for him. It really was a life-threatening situation, but it was ethically and philosophically complex because, there was this alignment, and there was this, question: is this is part of the human response, perhaps, to a perceived threat to the community? And so, but it was clearly not working, for him, and he was in danger. So I did treat him initially against his will, and this is a very interesting thing that can happen in psychiatry. As in much of medicine, there are cases where patients are initially not willing to go through with treatment, and there's a process with judicial oversight and so on that happens. But I ended up giving him an antipsychotic medication that has dopamine and serotonin receptor action. This was olanzapine, and it over the course of a couple weeks, it cured his mania. So this is it. So it was resolved. Let's put it that way. It resolved his state, and he returned to close to his baseline functioning. And follow up a year later, everything was superficially fine, or even his wife said he's better than ever. I remember that. I'll remember that quote forever. But the really interesting thing was that, and he was calm and sleeping and back to his typical state, but there was still this cognition that was not quite resolved. He still didn't understand why he had to be treated, or why I thought he had to be treated, and didn't understand that there was something that was not working so seriously that intervention was necessary. And I don't, here I am, I'm a fallible human being. I'm not saying with any kind of absolutism what was right or not. And in a way, that's the essence of what makes it so interesting.
Mm-hmm. Good. All right. So our Center for Translational Social Neuroscience is interested in social behavior and understanding disorders like autism. And so one of your chapters is about autism, and you make an interesting point, an observation about, people with autism tend not to look into the eyes. And maybe that's because they have fear. Some people say fear of looking into the eyes, aversion to looking into the eyes. And you actually, you talk about maybe what your understanding is of why people with autism tend not to look into the eyes. And then you also talk about insights that you've gained from animal models with optogenetics. Can you just discuss that a little bit, of your thoughts about autism?
Yeah, well, it's great. I mean, you're, of course, a pillar of the social behavior world. It's so great to talk to you about this, and your insights are probably far deeper than my own. But social behavior is something I'm very interested in. In my clinic, I see patients with autism spectrum disorder and also depression. Those are my two main clinical interests. And so I've had adult autism patients along the way the whole time. And what can you do? There's not a medication for autism, but I can help with their comorbid symptoms. A lot of the patients with autism are very anxious, and the world is a very unsettling and confusing place, being such a social world in a highly connected world, and there's a lot of pressure on to say and do the right thing in social settings, and it's very hard for them to predict what's the right thing to say in a social setting, and it's very anxiety provoking. And so I can help them with that. I can reduce panic attacks. I can provide medications, even simple medications like benzodiazepines to reduce anxiety. And so I had a patient, and the social behavior chapter, really, there are two patients that form the core of this, one with an elevated social state and one with a reduced social capability state. Both people that I saw, the autism spectrum patient had he's called Charles in the chapter. He was verbal enough so he could talk and communicate, but he had very pronounced autism symptoms, including very striking eye contact aversion. So he would always, to this day, he always looks away, he'll make brief eye contact, but then he'll look away, to the floor, to the side. And this is very common in autism, but it's pretty rare to get really into a deep discussion with a patient about why. And I've been treating Charles for a couple of years. He had panic attacks that were triggered by, a number of things, including social interaction. And I was able to treat that. And he'd gone from having debilitated panic attacks at work to having none at all. And that was resolved. It was a cure of the panic disorder. But it didn't affect the eye contact thing at all, not one tiny little bit. And so, I had a pretty good rapport with him over the years. And so eventually I asked him, what does it feel like when you make eye contact in that moment and then you immediately look away? Is it fear or something else? And he said, no, it's not fear. I'm not afraid. And it's not anxiety. It's not fear or anxiety. So we talked a little bit about what it was, and his description was so vivid. It mattered so much to me to be able to hear this. Autism patients often are not verbal enough to give clear descriptions, but he could do this. And he said, when I do look into your eyes, it makes me aware that I have to track and do something, and it's too much. There's too much information coming in, and it overwhelms the rest. And that was such an interesting descriptor, that there was too much information, that it was too rich, and that it overwhelmed the system. And there's so much about that is intriguing from the neuroscience standpoint. And it made me think, if you think about the social interaction, as you for sure have, and probably everybody else on this Zoom, there's a lot of information coming in the social interaction, but it's with a special quality. It's constantly changing. So other forms of high content information are fine for people on the autism spectrum. In fact, the richness of dealing with information and data often is fine, as long as it's not constantly changing. So as long as it's static, it's actually very, very appealing. But it's the rapidly changing information rate that is a challenge. And there's not much more than the social interaction for constantly, high rates of information per second.
It's a very dynamic situation. You've got to read things that are not always intuitive, and facial muscle contractions and expressions and things like that. And
yep, models constantly changing with each millisecond. You reshape the model, their predictions, and your analysis of the past. And so then this concept of information overload was interesting. And so in mice, what can you do? Well, first of all, we know if you make autism spectrum disorder mutations in mice, very often you will see social behavior deficits in the mice. And mice are social, as you well know. They do prefer to be with each other. They have engaged, investigation, and it's rewarding. And those behaviors are disrupted if you make certain, highly penetrant autism spectrum disorder mutations. And so it's studyable in mice, and moreover, one can create, using optogenetics, one can create very cell and circuit specific changes that cause or correct these deficits, as we found. And what seems to work very powerfully is shifting the balance between excitation and inhibition at the cellular level in the brain, and particularly in prefrontal cortex. This is an area where the balance between excitatory and inhibitory cells is very important, and it's poised on really what may be a criticality threshold. And if there is an imbalance in the relative strength of the excitatory cells that excite their neighbors, or the inhibitory cells that dampen down the activity of their neighbors, if those two cell populations are out of balance, you can create very marked changes in the information processing of the cells in the circuit. And we could actually show this optogenetically, that if you elevate the excitability of the excitatory cells optogenetically, you reduce the bits per second that can flow through that cell. And so you really can, if you elevate the excitation to inhibition ratio, you do overwhelm the information carrying capacity of the symptom in a very precise way. But it's not just bits per second, it's also behavior. If you do that same optogenetic manipulation, you elevate the excitability of the excitatory cells, as we found in 2011, you create profound social behavior deficits in mice that can be corrected by restoring the balance. If you increase the excitability of the excitatory cells, you get that dysfunction. But then if you bring up the inhibitory cells, you correct that. That's all in wild type mice. A few years later, in 2017, we did that whole set of experiments optogenetically, but in autism gene mutant mice. There's a gene called CNTNAP2 that causes, in humans and mice, social behavior deficits. So these are mice starting from an impaired social behavior baseline. And we did the opposite manipulation. We were able to take them, and these are mice that, if you actually look at their behavior, they're at baseline deficient in their social interaction. So if we, presuming that what's going on is that their excitation is elevated at baseline relative to inhibition, we found we could correct their social behavior by elevating the inhibitory cell activity to restore it back, the balance back to where it had been. So this was a case where optogenetics gave us this causal circuit level understanding that there's a balance at the level of cells that is critical for carrying out the complexities of the dynamic social interaction. And it's just, for me, it's just an incredible privilege to have also, in my experience and interaction with a patient like Charles, who made this all come together in such an interesting way.
Yeah. It's amazing how, you can change behavior and sort of mimic disorders, or just by shining light into neurons. And now you can control specific neurons. And in a later chapter, you talk about where you can control estrogen receptor neurons in the ventral medial nucleus of the hypothalamus. And you can just shine a little light on certain cells, and then the animals show aggressive behavior, and they'll attack. I have also that video that I use in classes. They'll attack gloves, and so it's just incredibly powerful. So we talked about these genes being sort of maybe providing useful characteristics, and and maybe we all have some complement of these genes. In fact, when I was reading your book, and I came across one of your descriptions of one of these disorders, and it struck me as very intriguing. And I read the passage to my wife, and she says, "Oh my God, that sounds like you." And I'm not going to tell you which one it was, but so I just made me wonder, as you were writing this book, or for these different disorders, do you ever think about yourself, and where do you fit on some of these spectrums?
Yeah, well, I absolutely do. I think most people who are introspective, I think that's a pretty healthy thing to do, to look in and see your own variants, where your unusualness comes from. That insight can help. First of all, being unusual can be helpful. You can maximize, the strength that comes from that, and also can help you if there is a weakness that comes with it. It helps you, mitigate that. And I've thought about that a lot. And actually, in the mania chapter, I make reference to, I had a crisis event that might have put me into what we call a hypomanic state. That's not quite mania, but it's close to, not requiring hospitalization. And I talk about that in that chapter. Then this is an amazing thing that happens in the course of writing. And by the way, anybody out there who is thinking about writing, as I'm sure many of you are, just get going. You don't know where it's going to take you, and you don't know what you're going to learn, and you don't even know what the overarching theme is going to be until you're done. So you got to start writing. Don't worry that you don't know even what the point is, because the process will tell you. It was only after I had completed, writing the book and looked at it in its totality, and I looked at the stories I'd chosen, the ones that mattered, and there were themes, and in particular, one theme that I'm also going to be a little coy, just like you, where I'm not going to say exactly what it was, but in the epilogue, I talk about this. The process of writing it really revealed a lot about myself, what really matters to me, what I'm most afraid of. And wow, that was that it wasn't even clear until it was done. So yeah, if you want that level of insight, which I don't know if people want, but you can get it if you can make it through to the epilogue.
Yeah, so the book, well, so we talked about autism. Something just popped into my head, and about autism and variation among us, and what some people being special, and we're just diverse in terms of our social skills. You got politicians can be very, very highly social. And you talk about one of the, in that chapter, someone who is very highly social, almost Williams syndrome-like. Then other people are very much more systematizing, focused on objects, and maybe that diversity creates advantages for us. And I remember Temple Grandin was visiting us here a couple of years ago, and she made the comment that, you know, maybe Einstein was possibly autistic, and Tesla was autistic, and maybe the person that 100,000 years ago that sat around and hit two stones together over and over and over again until fire was created may had autistic characteristics. And so that's a very interesting idea.
Yeah, I mean, if you look at what we're able to do as human beings, you know, so much of it depends on our diversity and on traits that, if we were all that way, we would not be functional as a species, you know. And it really is quite a remarkable thing to think about. And this comes up in essentially every psychiatric disorder. This is a theme in the book. This takes nothing away from the reality of the suffering, the reality of the dysfunction, the need for help, the need for treatment, the morbidity and mortality that comes from that. All that is not diminished by, though, this you know, realization that this you know, neurodiversity, if you will, is a real strength of our species, and maybe essential to our survival. Repetitive action, so fundamental. You know, cleaning. We're talking about the OCD spectrum now. You know, the cleaning without a reason why, without a detailed understanding of microbial, you know, biology. Very obviously some immense potential adaptive qualities to that. Social diversity, the range of people's ability to either engage with large groups and collect all the information, fuse it into a meaningful whole, and to take action in all that multidimensional state, to reduce it to dimensionality down in ways that matter, and to take action along the dimension that matters. That's obviously an incredibly powerful skill, and yet that may dwell in tension with another incredibly powerful skill, which is to look at a static, unchanging system with its own immense complexity. Take the example of Einstein, the incredible complexity of cosmology and physics and the required mathematics that undergirds it. Fortunately, that's not changing. You can look at it, you can take years and look at the same mathematical problem from every different angle, confident that it's not changing every 100 milliseconds like a social interaction.
Right.
And the ability to do that is, of course, of vast value to humanity as well.
Yeah, I want to take a time just to talk about a couple of things that were in your epilogue. I mean, the other chapters, you go into schizophrenia, which was really an amazing description of a person with schizophrenia, anorexia, borderline personality disorder in the other chapters. But let's skip to the end. I think you made some really important points about the value of basic neuroscience. Sometimes, it's hard to convince the public that, studying an algae or a vole or a mouse has anything to do with, curing disease. But your work is really a great example of how basic science funded by the government, leads to discoveries that provides tremendous insights into human biology. Can you just reflect on that a little bit?
Yeah, I mean, none of what we did would have been possible without, the purest of pure basic research, with literally no conceivable thread drawn or drawable to anything medical or even relating to human beings, to mammals. 150 years ago, a Russian botanist was studying single-celled algae and noticed that they moved in response to light. So it was plant behavior. Turned out, we now know, 150 years later, that was done, that algae do that with a channel rhodopsin, which is the workhorse of optogenetics. But it was discovered by a botanist 150 years ago. All our understanding of ourselves and of animal behavior comes from, you know, a botanist studying plants. How amazing is that? And there's literally no way you could possibly have predicted that. And then 50 years ago, Dieter Osterheld, who shared the Lasker with me, and Peter Hegemann, he was just studying archaebacteria, ancient microbes that live in high salt environments. And he was purifying their membrane. He was just interested in how these microbes work. And he noticed a bizarre color change when he changed the solvent that he was prepping the archaebacteria in, and that led to the protein level identification of this microbial opsin family. And so, you know, all of our understanding now and how it relates to psychiatry and maladaptive behavior, but also adaptive behavior, it all stems from this absolutely undirected, curiosity-driven exploration of the natural world. And so that's I think it's there are other examples of that, of course, but this is a particularly good one.
Mm-hmm. And the other thing that I think is so great about the book is that, and inspiring to people that are doing research in the lab, is that, you know, sometimes we just forget about the importance of what we're doing in the experiments. But, you know, at least for me, it made me realize, you know, how I can sort of think about what I'm doing in the lab in the context of what it's telling us about humans. I mean, we all write these things in grants, but I would encourage, you know, people that are doing lab work, students to and faculty to you know, read the book, because it's inspiring, especially, you know, people going to the medical profession. Sometimes they can be disillusioned about the process of medicine today.
Yeah. Well, you know, we all know science is hard, medicine is hard. And the combination is particularly hard. And the way the world is changing doesn't make it easier. Medicine becomes more codified. There's less room for creativity often, it seems. The ups and downs of you know, academic science life are, you know, are sometimes hard to make it through. And I've experienced those times, as we all have. There were, and I talk about in the book, times when I didn't think I could make it through, when I almost you know, left. I had some hard times as a single father, where it didn't seem as though I could make it through in any way. And but then, you know, the bigger picture, sometimes that we all sometimes lose track of, is so inspiring and exciting. And the things as neuroscientists that we are approaching are as big and interesting as anything in the universe. They really are. And I get to this in the epilogue as well. There are very deep, even philosophical questions that are now, you know, they're accessible now with modern neuroscience. And so we're at a really, you know, truly exciting time. There was, you know, I'll give one example. There was a Harvard philosopher slash mathematician named Hilary Putnam. He was still at Harvard when I was there. He was interesting. He started as a mathematician and then went to philosophy. So it was an interesting path. And he concocted a sort of a parable that he called the you know, evil scientist controlling a brain in a vat. I think he had an unwieldy title for it, but it was a nice sort of parable, because he said, what if, you know, you had a brain and there was an evil scientist, he actually said evil, who could interface with all aspects of this brain and provide thoughts and memories and so on. And he probed, you know, what the philosophical implications of that would be. But it was very, very broad and didn't map very well onto anything tangible. But now there's some really interesting things that modern neuroscience have brought us to. And one of them is this, and it comes back to what I mentioned earlier, this holographic stimulation of individual cells and creating percepts and naturalistic behavior patterns. There's this very interesting question. If you could create a single cell resolution activity pattern that wasn't truly naturalistic, that matched what a natural stimulus would do, then if you did that, would the organism truly feel the same subjective sense inside? Beyond the whir of information processing, would the subjective sense be the same? If I show you an incredibly rich, deep color red, sure, there's information processing, you register red, but also you have a feeling about it. Would that feeling still be there? And if the answer is yes, or the answer is no, you get into a paradox either way. And the progress of neuroscience is actually bringing us up to the border of this very interesting philosophical question.
Right. All right. Well, we're ending our time now. I want to thank you so much for taking time to talk to us. And, I want to recommend, the book. It's Christmas time, so think about for Christmas for yourself. I'm glad I read it. It was really amazing. And or gifts for family members. If you're a grad student doing neuroscience and you want your parents to know why you're doing neuroscience, this is a good way of doing that. And we're actually, for those of you who registered online beforehand, Karl was nice enough to sign 25 books. So stay tuned. If you're your name is drawn, you had to have registered and attended, then we'll be in contact about the book. So thank you so much, Karl. And sorry, everyone, we don't have time for questions, but we wanted to have as much time with Karl as possible with his thoughts. Thank you.
Thank you. It's been a lot of fun. Shoot me an email if anybody has any questions, but thank you, Larry. This was really amazing, and I appreciate it.
Okay. Thank you. Bye-bye, everybody.